Now we're going to switch over to talk about a different kind of analysis to look at gene expression, that's single cell data. I had 15 minutes for this. We're running short on time, so I might go a little bit over, but then we can go break into our break. It's the last day. I think we can do that. I'll do a quick primer on single cell data just to make sure we're all on the same page, and then we have an exercise to explore some of the single cell data in ViewPathDB. The exercise specifically is looking in TriTripDB, but of course, as we add more and more single cell data sets, the same tools and functionalities will be available to you in those resources. So for single cell data, just start with some I'm going to zoom out a little bit, and we're going to talk about the systems that we all investigate. And this is, we're looking at a brain made up of Legos here. And systems are obviously complex. We know that. They're spatial. They have functional and temporal dimensions, right? Different parts will have different functions. And of course, they change over time. Of course, in our case, in many instances, for many of us on this workshop, we're interested in systems that are infected, and so and whether this is an insect that's carrying a parasite, or it is a human brain or mouse brain that is infected with, in this case, Toxoplasma oocysts that are sitting in Toxoplasma brachioides or cyst forms, cysts that are sitting in the brain. It doesn't really matter. It's a system that is infected and that's carrying these pathogens. And so we can take these systems and analyze them in different ways, right? We're all familiar with these. We can sequence the DNA of the host and the parasite and explore them, or the host and the pathogen, and explore the DNA. We can assay the transcriptomic data using different technologies, including our RNA sequence data, and we can then analyze this RNA sequence data in different ways. We can assay the proteomics, we can assay the metabolomics, and you can imagine there are other types of analyses that you can run on this system. The big thing about what we've been doing so far in all of our analyses is that it's typically bulk analyses. You're taking the entire brain in this case, and you're mashing it all together. You're taking all this beautifully differentiated sections of the brain, and the parasite, with its big effort and specialization, went and localized in a different specific compartment in the brain, and you've mashed it all together, and you've then sequenced it all, right? So it's not bad. You're still getting a lot of information there, but you're also losing a lot of information. You're losing all the information about the specific cell types that these parasites are in, and potentially where they're located and so forth. Single cell omics technologies allow you to separate these cell types, including the infected ones, and find out in a bit more detail not only the changes in gene expression, but also which cells are more similar to each other and what is similar between these cells that makes them unique and different. And then obviously gets at different aspects of the biology, or more detailed and fine-tuned aspects of the biology. So obviously, these are some of the questions you can ask. Which cells are infected? Obviously, that's clearly here. You can see that the green and these purple and pink cells are infected. What is changing in the infected cells? So if this was a time course, not only can you tell which cells are being infected, but also in those specific cells that are being infected, what is happening during the infection process. You can even ask what's happening in these uninfected cells, right? So during the same infection, is something changing in uninfected cells? You can even ask, well, what about the cells that are close to these infected cells? Is anything changing in those cells? For example, cell types of the same cell type of the infected ones that are non-infected. And you can imagine this list of questions can go on and on and on, but being able to separate it down to the single cell layer gives you these options. I think most of you are familiar with this, but I think the key aspect of single cell omics is the ability to use these microfluidics to basically put a single cell into a single droplet. And in addition to having a single cell per droplet, you also are able to integrate all the enzymes and everything that you need to do your your chemical reaction. So you're, in essence, each one of these little droplets is a test tube that includes a single cell with all the components to do your assay in it. And so ultimately, in your individual droplets, you're doing basically an RNA sequence for single cell RNA sequencing experiments. And then after that, it becomes a computational um, technique to be able to tease out and figure out, well, what are the cells, what's happening in them, and what's going on. And single cell data is highly dimensional. So they are, in the case of what we're talking about here, you will collect thousands of cells, and each cell will have thousands of genes whose expression is changing in a particular experiment. And you may also have time points, of course. So the dimensionality is really mind-boggling. And also, one important thing to consider is that each cell is going to be different. So not all cells are created equal, and what you collect from each of these cells in terms of the genes and the expression pattern of those genes is going to be variable. So you will get many genes across the entire experiment, but so from all the cells, you might have an expression value for all the genes in your that are expressed or changing. But from each cell, you're going to only have a small subset or a subset of these genes. It's getting better and better, but typically you get a couple of thousand, maybe or less, expression changes per cell, and those are going to be different between the cells. And so that causes problem. It's a very highly dimensional data set, and there's potentially a lot of noise in there. And you have to figure out how to tease all of that stuff out from these experiments. And so what people typically do is they try to do something called dimensionality reduction. And you're probably familiar with many of these types of methods, including PCA, which stands for Principal Component Analysis, the T-SNE analysis, which is a T-distributed stochastic neighbor embedding. I never remember this. And then finally, the UMAP, which in the last few years has become sort of the more popular version of this dimensionality reduction, and that's the uniform manifold approximation and projection. Each of these have their advantages and disadvantages. You'll have people who swear that UMAP is the best technique possible, then other people come back and say, no, UMAP is horrible, T-SNE is better. And then other people say, well, actually a PCA is really, the best approach to doing this dimensionality reduction and trying to figure out what is happening in your data. It really depends on what questions you're asking and what you're trying to find out. And there are some differences between these methodologies, including whether the axes mean something. In PCA plots, you're actually teasing out the cell types based on basically a principal component, basically. And so most of your variation in your cell types are going to be due to maybe two or three, maybe one or two effects or conditions that you'll be able to tease out here. In these other methods, you are doing more of a clustering. It's typically non-linear, and then the difference between UMAP and T-SNE, there are many different statistical differences which I won't be able to explain to you, but T-SNE is much more computationally intensive to do, and so often people skip the T-SNE and do a UMAP because it's less intense computationally. But there are potential reasons why you would want to do T-SNE versus UMAP and so forth. But ultimately, people end up doing a PCA first, and then they will then extend this out to other methods of visualization. And specifically for visualization, you can see that a UMAP looks prettier than a PCA plot. But again, that's all depending on what you're interested in. And we can have further discussions about this later on. And then the last thing I wanted to point out, and again, I won't go into the detail, is that in addition to just looking at the being able to tease out the cells that are infected or even cells that are just changing over time, there's also spatial single cell transcriptomics now coming to light. We're already have some data sets that are available. There's one sort of I guess, quasi spatial transcriptomic data set in PlasmoDB for liver cells. But I mean, it's pretty cool that we can start doing these things, and you can imagine the application of this and the amount of data that's going to be generated over the coming years. And we're excited about integrating that data. Okay, so this is all I was going to say about single cell data. And I'm sure there are lots of questions which we can discuss further in the breakout rooms or here afterwards. Any other instructors, anything else you wanted to point out or highlight that I did not highlight?
Don't think so, except to say that, as Omar said, there's much more detail here. All of you will probably have colleagues or informaticians locally who can provide more statistical information on what it is that's looking at. Most of the instructors are probably happy to answer some of these questions in their breakout groups, or you're welcome to ask them later on, or click on the Contact Us and we can provide you with some answers.
So for the exercise, just to orient everybody, we're looking at this experiment from Briggs and colleagues from Richard McCulloch's lab, where they looked at differentiation in Trypanosoma brucei. And so these parasites have, like all parasites and organisms, have different developmental stages. They have different names, slender versus stumpy. Simply the slender are cells that are proliferating, so they're growing and replicating. And then at some point they differentiate into these stumpy forms, which are a little bit more quote unquote dormant, and those are the ones that get picked up by insects for transmission, being able to go through another developmental cycle there. And so in this experiment, they did single cell on the differentiation cycle, and they were able to tease out a potential development regulator, which is listed here, the gene ID. The second part of this exercise, which we listed as optional because I don't think we're going to be able to get through it today, but this experiment was two parts. One part was looking just simply at wild type cells and looking at their differentiation and trying to tease out genes that are potentially important for regulating differentiation. And then the second part was that they did an experiment with a mutant where this gene that they identified was knocked out. And then they were able to then identify, identified as a key component in regulating differentiation using single cell data.